The third line of the body's defense, which is also known as the last line of defense of the body, is the specific adaptive immune system. This system of cells is what is properly described or defined by many medical experts and educators as the immune system. The term immune system is equated with the adaptive defense system only. However, we know now that the innate and adaptive defenses are deeply intertwined. As pointed out in the first video lecture, the immune system is a functional system rather than an organ system in an anatomical sense. Its structures are a diverse array of molecules plus trillions of immune cells, especially lymphocytes, that inhabit lymphoid tissues and circulate in the body fluids. When it operates effectively, the adaptive immune system protects us from a wide variety of infectious agents as well as abnormal body cells. When it fails or is disabled, devastating diseases such as cancer or AIDS result. As we have learned from the past video lecture, the nonspecific innate defenses includes surface barriers and internal defenses that are not specific for any particular antigen. The specific and adaptive immune system, on the other hand, is very specific on what particular antigen it will bind to and attack, and is conventionally classified into two parts: the humoral immunity and cellular immunity. Humoral immunity primarily involves the B lymphocytes, and cellular immunity involves T lymphocytes. They have distinct mechanisms of actions, but most often than not, they overlap. It must be emphasized, however, at this point, that innate and adaptive defenses are inextricably intertwined mechanisms. Both B and T lymphocyte precursor cells originate in the bone marrow. Lymphocyte precursor cells destined to become T cells migrate via the blood to the thymus and mature there. B cells, on the other hand, mature in the bone marrow. During maturation of B cells and T cells, each lymphocyte synthesizes multiple copies of particular receptors on its plasma membrane that enables them to recognize specific foreign molecules of potential invaders. Thus, they develop immunocompetence. Lymphocytes that have receptors that can bind to any molecule in the body and normal body cells are destroyed by clonal deletion or disabled by clonal deactivation. This way, the immune cells cannot attack its own cells. This allows immune cells to possess self-tolerance. Immunocompetent but still naive, the lymphocytes leave the thymus and bone marrow, and they seed the secondary lymphoid organs and circulate through the blood and lymph. When a lymphocyte antigen receptor binds to antigen, that lymphocyte becomes activated. Presentation of antigen that activates B or T cells also requires the antigen being bound to a class of plasma protein called major histocompatibility complex, or Mhc proteins. The Mhc proteins restricts the ability of lymphocyte receptors to recognize antigens. In this way, the immune cells become very specific for that particular antigen. Any element, whether fragments of foreign cells or whole foreign cells, that trigger response of the immune system is called antigen. The processes by which the specific immune system is brought into action is when specific immune cells encounter and recognize an antigen, become activated, and then respond to the antigen in a coordinated manner. The main group of cells that carry out the specific immune response are only two subpopulations of lymphocytes, called B lymphocytes or B cells and T lymphocytes or T cells. Encounter and recognition is a process whereby receptors of lymphocytes bind to a fragment or whole foreign microorganisms that are presented by a particular protein, major histocompatibility complex, or MHC proteins. Upon activation of the B cells by cytokines and antigen presentation by white blood cells, the B cells transform into plasma cells that secrete antibodies specifically designed to bind with that particular antigen. Plasma cells are not really distinct cell types, but are cells that differentiate from activated B lymphocytes after they encounter antigens. The activation of B cells and antibody secretion constitute the humoral immune response. Activation of B cells and T cells, or helper T cells and cytotoxic T cells, require antigen receptor and Mhc protein binding. When helper T cells are activated with antigen, they secrete specific cytokines that coordinate the immune actions or immune responses of plasma cells and cytotoxic T cells. Activation and direct attack of cytotoxic T cells on foreign cells or substances constitute the cellular immune response. So, the action of B cells turning into plasma cells that secrete antibodies are called, or rather is called, the humoral immune response, and that of the action of cytotoxic T cells directly on foreign cells or infected cells constitute the cellular immune response. B cell receptors and plasma cell antibodies are Y-shaped proteins that constitute the family of proteins known as immunoglobulins. Although the B cell receptors are structurally similar, only secreted immunoglobulins from plasma cells are technically classified as antibodies. When bound to plasma protein of B cells, they are simply called immunoglobulin receptors. Immunoglobulins have a stem called Fc portion, and the upper part of the protein prong is the antigen binding site, which is the most variable part that allows millions of different receptors and antibodies to bind to any kind of antigen one can encounter in a lifetime. Antibodies secreted by plasma cells are very specific for that antigen they have encountered from either macrophage, B cells, or dendritic cells. After their differentiation from B cells, plasma cells produce thousands of antibody molecules per second before they die in a day or so. There are 5 different classes of antibodies. The most abundant of these are the IgG or gamma globulins. The antibodies bound to antigen on the microbial surface do not directly kill the microbe, but instead link up the microbe physically to the actual killing mechanisms such as neutralization, agglutination, precipitation, opsonization with complement proteins, phagocytosis, inflammation or enhancement of inflammation, and cell lysis by natural killer cells. The linkage not only triggers the attack mechanism, but ensures that the killing effects are restricted to the microbe. T-cell receptor cannot combine with antigen unless the antigen is first complex with a certain plasma membrane protein. These are the major histocompatibility complex proteins, or MHC proteins, named after the genes that encode for them. There are 2 classes of MHC proteins, 1 and 2. Class 1 MHC proteins are found on the surface of virtually all cells of the body, except erythrocytes. Class 2 MHC proteins are found mainly on the surface of macrophages, B cells, and dendritic cells. Under certain conditions, other cell types are induced to express class 2 MHC proteins. The diagram shown here summarizes and simplifies the interactions of nonspecific defense cells with that of the specific immune cells in launching the defenses of the body, particularly the cellular immune response and the humoral immune response, also known as antibody-dependent immune response. It starts with the encounter of the antigens by nonspecific cells or specific immune cells. When the antigens are encountered by the nonspecific cells like dendritic cells in the periphery of the body, the dendritic cells phagocytize the antigen and presents this to specific T cells. antigen presentation to these specific T cells activates the T cells, causing them to undergo mitotic proliferation called clonal expansion. In other words, this activation causes the activated T cells that have encountered a specific antigen to multiply by mitosis, multiplying the activated T cells that can respond to the same antigen. And in the process, the CD8 T cells give rise to cytotoxic T cells. The cytotoxic T cells, once activated, directly attacks infected cells or the foreign microorganism, such as by inserting transmembrane proteins called perforin on the plasma membrane of infected cell or microbe, causing them to leak out of cytoplasm. This directly kills the infected cell or foreign microorganism. Meanwhile, the activation of the CD4 T cell also causes them to undergo clonal expansion, multiplying the activated CD4 T cells that can react or can respond to a specific antigen, and in the process give rise to helper T cells. The helper T cells coordinate immune responses of the body, and so they send signals such as cytokines to various cells, like the activated CD8 T cells are further activated by signals coming from helper T cells, or helper T cells can also send cytokines to non-specific cells like natural killer cells or macrophages that participate in direct killing of the antigen, foreign microorganism, or infected cells, helping the cytotoxic T cells to kill the foreign cells or infected cells directly. The helper T cells can also send signals to the B cells as they are activated by the encounter of antigen. It enhances their activity and transforms them into antibody-secreting plasma cells. The B cells, the activated B cells, can also send signals to helper T cells that can also coordinate the immune responses in the body. And so with this, you have launched the cellular immune response and the humoral immune response with activation of the different T cells and B cells. Meanwhile, when the infection has already been contained, a few of those activated T cells and B cells remain in the blood throughout life, so that when the same antigen is encountered once again, the immune response can be quicker than the previous one, and the defenses of the body becomes more efficient with memory T cells and memory B cells. In most cases, the non-specific defenses and the specific immune cells are very efficient barriers against pathogens and even against our own cells that have malfunctioned. Sometimes, however, the immune system backfires or become insufficient that might cause harmful responses instead. Immunodeficiencies are congenital or acquired conditions that impair the production or function of immune cells, or certain molecules such as complement proteins or antibodies. The most devastating congenital immunodeficiencies are a group of related disorders called severe combined immunodeficiency syndromes, or SCID, which result from various genetic defects that produce a marked deficit of B and T cells. The most devastating of the acquired immunodeficiencies is acquired immunodeficiency syndrome, caused by HIV, or human immunodeficiency virus. This cripples the immune system by interfering with the activity of helper T cells. Occasionally, the immune system loses its ability to distinguish friend or foe from foreign antigens, When this happens, the artillery of the immune system, like friendly fire, turns against itself. The body produces antibodies or autoantibodies and cytotoxic T cells that destroy its own tissues, and this produces the autoimmune diseases. The T cells, cytotoxic T cells, can destroy its own tissues, like myasthenia gravis, multiple sclerosis, and type 1 diabetes. Hypersensitivities, also known as allergies, result when the immune system damages tissue as it fights off perceived threats which are actually harmless to the body. So here we end chapter 18, section B, about the specific immune system. Be ready for quizzes, especially from previous chapter and to this chapter. And so we end here with our quarter, and be ready for the final exam this week.